Every tablet faces the same first challenge. Before it can heal, it must dissolve. But how do we know it's dissolving the way it should? That's where pharmaceutical dissolution testing comes in. Pharmaceutical dissolution testing measures the rate and extent to which an API is released from a dosage form into a defined medium under standardized conditions. In plain terms, it tells us how fast drug becomes available for absorption, a key driver of bioavailability. And remember, it's not only tablets that undergo dissolution testing. Capsules, suspensions, powders, oral films, and other dosage forms all can be evaluated to understand how efficiently the drug leaves its formulation and becomes ready for absorption. So why do we test dissolution in the first place? First, for quality control, dissolution ensures every batch performs the same way and that the medicine you take today behaves just like the one you took last month. Second, during formulation development, we use it to fine-tune prototypes, adjusting excipients, particle size, or coating until the release profile is just right. Third, it's the cornerstone of regulatory bioequivalence, as dissolution can act as an in vitro mirror for what happens in the body, especially for highly soluble, highly permeable drugs. And finally, there's stability. Over time, different factors can quietly alter a formulation. By tracking dissolution, we catch those changes before patients ever do. Now, let's peek under the hood. What's actually happening when a drug dissolves? Imagine a tablet dropped into fluid. Drug molecules begin to escape from the surface and move into the surrounding liquid, spreading from areas of high concentration near the surface to lower concentration farther away. This entire process is described by the Noyes-Whitney equation that captures how a solid drug becomes a dissolved one. Here's what that means. D is the diffusion coefficient, a measure of how easily drug molecules move away from the solid surface and into the surrounding liquid. A is the surface area of the exposed solid. The more surface that touches the liquid, the faster molecules can leave. This is why particle size reduction has such a dramatic effect on dissolution. CS represents the solubility of the drug at the solid surface, while C is the current concentration in the bulk solution. The difference between them is the driving force for dissolution. The greater this gap, the faster the drug dissolves. Finally, H is the thickness of the diffusion layer, a thin film of stagnant liquid around the particle. A thinner diffusion layer, achieved by stirring or increasing hydrodynamics, allows molecules to escape more easily and speeds up dissolution. The standardized framework for dissolution testing is established in the USP Chapter 711, which defines the apparatus, test conditions, and acceptance criteria used globally to assess drug release from dosage forms. In practice, dissolution testing is conducted using one of several official apparatus, each designed to simulate specific physiological or formulation characteristics. Apparatus 1, known as the basket method, employs a rotating cylindrical mesh basket that holds the dosage unit. It is particularly suitable for capsules and tablets that tend to float or disintegrate slowly, ensuring consistent exposure of the surface to the medium. Apparatus 2, the paddle method, is the most widely used configuration, especially for immediate release tablets. The rotating paddle stirs the dissolution medium uniformly, providing reproducible hydrodynamic conditions. Apparatus 3, the reciprocating cylinder, is designed primarily for modified release and delayed release products. The apparatus moves vertically through a series of vessels containing different media, simulating the sequential pH environments of the gastrointestinal tract. Apparatus 4, the flow-through cell, allows continuous flow of dissolution medium through a packed cell containing the dosage form. This setup is ideal for poorly soluble drugs or dosage forms requiring sink conditions, as it maintains a constant supply of fresh medium. Later variants, apparatus 5, 6, and 7, adapt these basic designs for specific extended release or transdermal delivery systems. The dissolution medium itself is carefully selected to simulate physiological conditions within the gastrointestinal tract. Commonly used media include simulated gastric fluid, acetate buffer, and recently more of a bicarbonate buffer, and also phosphate buffer. For poorly soluble substances, surfactants such as sodium lauryl sulfate or SLS may be incorporated to more accurately reflect in vivo solubilization. Temperature control is critical. The medium is maintained at 37 plus or minus 0.5 degrees Celsius, mirroring human body temperature, and agitation speed is standardized to ensure reproducibility across laboratories. Several interrelated factors influence the dissolution rate of a drug, each aligning closely with the variables described in the Noyes-Whitney equation. To make them easier to remember, think of the mnemonic dissolve, each letter representing a key determinant of dissolution behavior. D is for drug properties, the physicochemical characteristics of the active ingredient, such as crystal form, particle size, and wettability, directly affect dissolution. I is for ionic strength and pH of the medium. Both can alter the drug's ionization state and solubility. For weak acids and bases, pH changes can shift dissolution rates according to the Henderson-Hasselbalch relationship. S is the surface area. The exposed surface of the solid drug controls how much contact it has with the solvent. A larger surface area, whether by micronization, amorphization, or improved wetting, promotes faster dissolution. The second S is the stirring speed. We discussed that agitation maintains uniform concentration in the bulk medium and helps reduce the thickness of the stagnant diffusion layer, H. O is the organic solvent presence or surfactant use. For poorly soluble drugs, small amounts of co-solvents or surfactants improve wettability and maintain sink conditions, ensuring continuous dissolution. L is the liquid medium volume. Adequate volume is essential to maintain sink conditions. That is, the ability of the medium to dissolve at least 3 times the amount of drug present in the dosage form. Insufficient volume can lead to artificially low dissolution values. And V is the viscosity of the medium. Increased viscosity slows down molecular diffusion, thereby reducing the dissolution rate. This factor becomes especially relevant when polymeric media or hydrogel matrices are used. The final letter E is the equipment type. Each USP apparatus creates distinct hydrodynamic conditions influencing the reproducibility and comparability of dissolution data. Choosing the correct apparatus for the dosage form is critical for valid results. Once the dissolution test is underway and samples have been collected, attention shifts from how the test is performed to how the results are interpreted. The first key measure is the Q value, the percentage of the labeled drug content that must dissolve within a specified time frame. For example, a typical requirement might be Q equals 80% in 30 minutes, meaning that not less than 80% of the drug should be dissolved at that sampling point. The second parameter is the dissolution profile similarity factor, denoted as F2. This statistical measure is used to compare the dissolution profiles of a test product and a reference, usually an innovator formulation. An F2 value between 50 and 100 indicates that the two profiles are sufficiently similar, and this is a critical consideration when demonstrating bioequivalence for post-approval changes or generic formulations. Finally, sampling times are selected strategically, often ranging from 5 to 60 minutes for immediate release products and extending longer for modified release systems. These time points are chosen to capture the entire dissolution profile, from initial disintegration to complete drug release. With the numerical data we just explained comes also the shape of the dissolution curve itself. Immediate release formulations show a steep, rapid rise, reaching a plateau near full release within a short time. Delayed release forms display a flat lag phase at first, no release in the acidic stage, followed by a sudden upward slope once the coating dissolves at higher pH, and extended release systems produce a gentle, gradual slope, reflecting slow, continuous diffusion of the drug over several hours. These distinct patterns visually confirm that each formulation is performing exactly as designed. And that is a wrap for today's video. One final question for you. Which apparatus have you used, basket or paddle, and at what RPM? Comment below and subscribe for more. And as always, till next time, stay fabulous wherever you are.